Hello everyone, and welcome to my very first class in my channel. And my very first class is about microbiology. I won't do any kind of introduction because I already did a video about the introduction of this channel, so I'll start by teaching you my very first class, which is about bacteria. First of all, I wanted to mention something that I told in my very first video, that I will provide questions at the end of the video or at the beginning of the video. So maybe it will appear at the end of the video. And as we know, like as a medical students, that if we question ourselves as a space repetition study process, that will increase our knowledge more, and it will also help to keep our memories in a long-term memory process. So let's start. So today we will learn about 5 bacteria. The first one is Enterobacter, the second one will be E. coli, third one will be salmonella, fourth one shigella, and fifth one will be yersinia. So in today's video will be a bit longer, so please bear with me, and I hope and I promise that you will benefit a lot from this video. So I divided my video into 3 sections. First one will be the features and also how it transmitted, like what are the treatments for these bacteria if someone get infected, and second one will be how we diagnose these bacteria, and third one I will provide the clinical cases, as I said in my introduction, that I will try to provide and relate the studies with clinical cases, so we will be able to learn more. So let's get started. So without further ado, we can start right now. So our very first bacteria will be Enterobacter. So as we know, Enterobacter, there are some bacteria which are in our normal microflora. So inside in the section of the Enterobacter, E. coli also comes. So first we will learn Enterobacter, the morphology. The morphology of this bacteria is gram-negative rods, because if we see under the microscope, we will see this kind of shapes of the bacteria, which is like a rod shape, and it is also a gram-negative staining. We will see those bacteria under the gram-negative staining. Then these bacteria are motile, like could be motile or could be non-motile as well. If those bacteria has flagella, those bacteria will be motile. There is one question is, if the bacteria is containing one flagella, so that bacteria will be more, sorry, I had a wrong shape. So this is a shape of the bacteria. If the bacteria is containing one flagella, so this bacteria will be more motile. And if the bacteria is containing multiple flagellas, then this bacteria won't be that motile as the bacteria which has one flagella, because we know the flagella actually helps as an adhesion factor, so it can bind to the host more properly. So if we get more flagellas, then bacteria will be bind to the host cell, so it won't move that much. Then we have the third feature, which is asporogenic, and the fourth one is this bacteria is encapsulated, and then this bacteria is facultative anaerobes, as we know, which means that this bacteria could survive with oxygen and also without oxygen. So 50-50 situations. And this bacteria actually grows in the raw meat, here we can see. And if we want to cultivate this bacteria into the labs, we have to use McConkey agar and also endo agar. And then next comes the biochemical properties, which is very important to know how this bacteria is working or how it's releasing toxins or everything else. So the features are these bacteria are catalase positive, oxidase negative, and it can reduce the nitrate into the nitrite. And if we use the API kits, we will be able to justify the biochemical properties more precisely. And then we have the pathogenic factors, which what kind of factors causes the diseases into the body. Now the factors are adhesion factor, as I said, like the flagellas, or it could be pili, and these pili actually bind to the host cells and causes an attachment with the host cells and also release toxins, and then it actually initiates all its mechanism, and eventually the cell dies, the host cell dies. And the major feature of Enterobacter is also for E.coli, it causes urinary tract infections. So in the hospitals, we will be able to see more urinary tract infection caused by E.coli or Enterobacter. Then the second option is the capsules, and this is the resistance factor, and it can actually invade the phagocytotic mechanism, which is caused by the host cell. Then there is the plasmid. We have this plasmid has three kinds of features, which is resistant plasmid, cold plasmid, tox plasmid. Resistant is as we have seen that it will get resistant against the host cell defense mechanism. And then we have the cold plasmid, which actually the main reason of the virulence factor, which will actually spread the toxins or the pathogenic chemicals. And there is the tox plasmid, and this is classified as exotoxin and endotoxin. So the exotoxin is secreted actively, and you can read here that the E. coli, it causes diarrhea, and because of the tox plasmid, it actually invade the enteric cells, like in the small intestines, and then it actually activates the adenyl cyclase, which is inside the cell. As a result, there is a camp inside the enterocytes, which is a chemical, and it actually gets activated and gets more higher, and as a result this mechanism releases more water and electrolytes in the gut lumen, and at the end it causes diarrhea. And the second toxin is endotoxin, and this endotoxin causes sepsis and also endotoxic shock, as we know that the sepsis actually happens when there is a bacterial elements in our blood. So this is another reason that endotoxin might cause the sepsis if we see the patient is having, like if we take the blood of the patient and watch it under the microscope, we will be able to like identify the bacteria and also the reason of the sepsis. Now the second thing I would like to share is the difference between the exotoxin and endotoxin. As I said in my introduction video that I will provide materials which came into the USMLE tests and also from the USMLE books. So this is USMLE step 1 material. So this one here I won't read it because I will share this note at the end of the video, so you guys will be able to learn more. So just please stop the video and just learn and see and read what's written in this video. Then I talked about that how endotoxin and exotoxin work. So we can see here, so the toxins inside the exotoxin bacteria, they carry those toxins inside the cells of the bacteria. So after some kind of mechanism, they will release the bacteria, they will release the toxins. But in case of the endotoxin, they will lyse the cell of the bacteria, and then those toxin will come and then it will bind to the host cell receptors. And from the host cell receptors, when they actually know that some foreign elements has been entered into the cell, they will release those TNF and interleukins to cause further mechanism like fever and also diarrhea. And then there is another material, which is how and what is the meaning of the endotoxins. So this is an abbreviation, so it's really easy to memorize what every alphabet means in the endotoxins. So last one would be shock. So these are the features of endotoxin. Then the N defines as neutrophil chemotaxis, and then then I defines interleukins 1 and 6. So this is specific that endotoxin only release interleukin 1 and 6. Then the X, so this endotoxin bacteria which causes, which contains endotoxin, especially from the E.coli, are extremely hot stable. And then we have it has O antigen, and also it has core polysaccharide and lipid A. So these are the three elements which the bacteria is composed of. Then the next element is TNF alpha, which is another reason of fever. If someone is getting affected by E. coli, and then there is an outer membrane of E. coli bacteria, which is responsible for the endotoxin release, because those outer membrane actually lyses, and then here you can see then the bacteria goes to the toxins, my bad. So the toxins goes to the host cell. And the last thing is it has nitric oxide. And the main important thing we need to know that endotoxin causes edema. And there are some more materials which actually came into the USMLE test, so you can just pause the video and can read it out, because I just said the same thing here in the endotoxin part. And then there are more USMLE contents here. So these are the bacteria which actually contains the exotoxins and also causes the harm in the host cells, which are they are actually, first of all, they inhibit the protein synthesis. And these are the bacteria here. And these are the toxins which they actually carries. Then the second feature is they also increase the fluid secretion, which is known as diarrhea. And as we know, as a treatment of E. coli, it would be giving electrolytes or IV fluids, because we are secreting fluid out of the body. So it is must that we need to hydrate the patient, so there will be more fluid inside the body of the patient and they won't lose more water. Then the third feature is they inhibit the phagocytotic ability, because as we have seen up there in the morphology that they are encapsulated and they have capsules which actually invade the defense mechanism of the host cell. And then we have fourth one, they inhibit the release of the neurotransmitters, and they also lyse the cell membranes, and they have super antigens, which are the cause of the shock. And here are the toxins, and these are the bacterias. If you make a table or make flashcard out of this content, then it will be more easier to learn. Then our second bacteria is Escherichia, or as we know, E. coli. Their morphology and biochemical properties and pathogenic factors are more similar to the Enterobacter, and also their symptoms and also the clinical symptoms and also the treatment are more likely same. So these are the features. You can just pause the video and can take some notes here, but the main thing I would like to emphasize is E. coli is the only bacteria which actually ferment the lactose properly and produces acid and gas. And here you can see the same plasmids, and these are the temperature which actually needed for the bacteria to grow in the human and also mammals. And here another USMLE content, which is the type 3 secretion. So in case of E. coli, we have 5 types of E. coli, so I will explain more further down there in the note. So there is a type 3 secretion. When the bacteria binds to the host cell, there are needle-like appendages coming from the bacteria, and these appendages are like needle-like shape body parts of the bacteria directly deliver the toxins to the host cell. So they don't need any other helps to deliver the toxins, like nothing. They just go to the host cell and inject something directly from their body, and game over for the host cell. Here, this is also gram-negative bacteria. Now comes to the classification of the E. coli. As I said, there are 5 types of E. coli. There is enteropathogenic E. coli. And then we have, okay, never mind. So there is enteropathogenic E. coli, there is enterotoxigenic E. coli, there is enteroinvasive E. coli, and then enterohemorrhagic E. coli, and which is the most common E. coli, and this is the serotype, which is also the most common serotype found in the human body, because many people ask, what does this O and H means? These are actually the antigen types. If you studied in the first year of medical school that in the bacteria there are in the bacterial cell we have 4 types of antigen. So this is O antigen, which is known as the capsular antigen. It actually describes the type of the capsule which is surrounding the bacteria. And then there is there's a mistake, that O antigen is the outer membrane antigen, what it is composed of. Then there is K antigen, which is capsular antigen, also what type of capsule it is. Then there is H antigen, which is important for the fimbriae. So these are the 3 types of serotypes. You will be able to see actually in each type of the bacteria. So these number depicts that what like how many antigens are there. So there are 157 number antigens is in the outer membrane, because we have so many antigens outside of the bacteria, so we just can count. So this is count as a number. And the flagella has, like, the fimbriae or flagellar antigen is depicted as H, because the antigen number, like the toxins which are in the in the flagella are like counted as 7 number flagellar antigen. That's what the whole number means. Then we have the aggregative E. coli. So just read out the like the table. So I made the table so you guys can make notes from it, and like we'll be able to memorize more effectively. And there is another USMLE content, which is the difference between the shigella and shigatoxin. As I said, like EHEC, which is E. coli hemorrhagic E. coli, enterohemorrhagic E. coli, it also secretes toxins, which is similar to Shiga toxins. That means the Shigella bacteria, the toxin that secretes is similar to the toxin which is secreted by the EHEC. So the only difference is from the Shiga toxin and Shiga-like toxins is it damaged the gastrointestinal mucosa, and as a result it also released the cytokines, and it causes the hemolytic uremic syndrome. That means we will see blood in the urine. If some patients are having like blood in the urine, so we will be able to identify that, okay, it's happening for the Shiga toxin, like direct Shiga toxin, not from the E. coli or EHEC. And the second one is Shiga-like toxins. It actually enhances the cytokine release, and it also caused the hemolytic uremic syndrome, but it does not invade the host cell. So in case of Shiga toxin, it actually invade the host cell, but the EHEC does not invade the host cell, so it doesn't directly affect the host cell. It actually uses a secondary mechanism to get into the cell. So here is a treatment. As I said in the beginning, that as we are losing more fluids, so the patient needs to get more electrolytes here, and also more vitamins and also fluid. So the patient needs to drink more water. And if some antibiotics is given to the patient, so as we know, if antibiotics is given more, we have like normal microfloral bacteria in our gut, so they might die as well. So these are the colibacterium and lactobacterium, which is given to the patient. So the normal microfloral bacteria doesn't die, and it also enhance the immune response. And then we have the salmonella. We are in our third bacteria. So there are 4 types of salmonella, which is salmonella paratyphi A, paratyphi B, and choleraezus, which is type C, and there is typhi, which is D. And this is the most important salmonella, which is the reason of typhoid. So just remember, this causes the main typhoid fever, and it's kind of dangerous if the patient doesn't get proper antibiotics. Here again, the morphology and also antigenic structures. Here you can pause the video and just read through this, but there's one special thing in the salmonella, which is this salmonella doesn't have any enterotoxin. That means no exotoxin. And also salmonella doesn't ferment lactose as E. coli, and it has type 3 secretion as E.coli, which is pretty similar to the E. coli. Here I wrote it, make sure you write it as well. And there is another difference, is salmonella doesn't have any spore, and on the other hand we have seen that E. coli has spore. And these are the main differences. And the main thing is we need to discuss is the diseases and also clinical finding. I have said that salmonella causes the typhoid fever and also enterocolitis, which is also known as food poisoning. If someone is having cramps, bad cramps in the stomach, or vomiting more after eating some roadside food, so it might be a food poisoning. We need to guess that unless the patient doesn't go to the hospital and check the stomach and also stool examination. Then the salmonella causes also bacteremia, which is also known as sepsis, and because the bacteria is in the blood. And eventually this bacteremia also can cause endocarditis, which is also really common. And carrying, that means the person which like who has this bacteria, might be a possibility that the person is carrying the bacteria. Also there is a possibility that this bacteria can be in the milk and also over the food, like the surface of the food or any fruits. So we need to wash everything so before we eat anything. And there is clinical findings, which is osteomyelitis. This is the most common cause, as we will be able to see in the hospitals that the patients have osteomyelitis, and it might be connected to the salmonella. And the meningitis and nephritis and also abscess, these are less common. Abscess as edema. And then the pathogenesis of the salmonella, how the pathogenesis occurs. So when the salmonella attacks the host cell, it colonizes surrounding the small and large intestines. So this is the main gate for these bacteria, and then it actually attacks the gut cells. And then they cause the inflammatory lesions in the small and large intestines. So there will be more interleukins T and F will be released in the intestines. And there is source of infection, how they cause the infection. I have made a table so it will be more easier to know. As I've said, chicken's meat, milk, because if we don't cook the chicken's meat properly, so there is a huge possibility that there is salmonella over the chicken meat. And also raw eggs and also milk. And on the other hand, there is a huge possibility that infected animals have bacteria in their tissue, in their meats or in the eggs. And here is the treatment comes for the salmonella is antibiotic ampicillin. So if you studied properly in the microbiology first year, so you will be able to know there's penicillin, which have 5 generations, and this ampicillin goes under the 3rd generation, as I remember. And these are the vaccines. So it's not a pandemic kind of bacteria, so there are no specific vaccines that like it's obvious to take in. So there is no like obvious of taking the vaccine. So you can take the oral vaccines, which is virulent. It's a live virulent strain. So the the virus will be the bacteria, I'm sorry, that would be bacteria. So the bacteria will be live, so and avirulent means the virulence factors and the toxins will be taken off in the lab, like by doing some uh, processes in from the live bacteria. We will take out the toxins and we will be able to take the vaccines, and as a result we will be able to immunize against that bacteria, which is salmonella. And the second vaccine is capsular polysaccharide vaccine, and also acetone killed vaccine. And yeah, this has the most effectiveness. I'm sorry for the type. Okay, let's back to the discussion again. What I was saying, that acetone killed vaccines, it has the most effectiveness, like as 75 to 95%, and every 3 years you need to revaccinate yourself if you're getting affected again and again, so you need to be careful more. And then our next bacteria is shigella, which is the fourth one. We have 4 species for that. First one is Shigella dysentery, Shigella flexneri, Shigella boydii, and Shigella sonnei. Again, the same morphologies. Here you can just pause the video and can read it, because I have explained most of the features. And again, this Shigella doesn't ferment lactose as salmonella, but on the other hand, E. coli does, and also Enterobacter does. But one exception is Shigella sonnei. It ferments the lactose, but slowly. So it takes 48 hours to ferment the lactose. So for example, if we get a patient and we are suspecting that the patient has E. coli, but at first we need to get the sample, then we need to transport the bacteria to the lab, then we need to cultivate the bacteria, and we need to observe that petri dish or the bacteria with the agar for 48 hours, because we can't just say, okay, the patient has E. coli. It could be also Shigella sonnei, because because of this exception that sonnei ferment lactose. And then we can see under the microscope the shape, and also we can look for the antibiotic susceptibility test along with the biochemical property test. And here is the virulence factor. So we have learned that in the salmonella it doesn't have any enterotoxin or exotoxin, but here we have in the shigella we have endotoxin, lipopolysaccharide, and enterotoxin, which causes dysentery. And because we have enterotoxin here, I wanted to say that, like, take a note that E. coli and Shigella are more likely will have same symptoms, except Shigella infected people will have high fever. In the E. coli, we have learned that it also shows diarrhea or dysentery as symptoms, so as Shigella. So do Shigella also shows the symptoms of the E. coli as dysentery. And there is the source of transmission. So first of all, it causes person-to-person contact. So if person is infected and person does not know, then there is a huge possibility that that person will be infecting other people as well. And also fecal oral contact, as we know, the meaning of this sentence is if someone is going to the toilet and he or she didn't wash their hand properly, and with that hand he's going to make food or eating any food, there is a possibility that he has or she has shigella or shigella type bacteria, or it could be E.coli if the hands are not washed properly. And another thing is if the water is not if the water is contaminated, and also through fingers and food and also flies, it could be transmitted by these three ways. So just be careful whenever you go to the washroom. And there is the mechanism of the shigellosis. So this is the most important mechanism of shigella bacteria. So here we can see in the diagram that you can see here, this is the shigella bacteria. So it cannot invade the cell directly, and then because it doesn't have, and here in the features you can see it doesn't have type 3 secretion. So in the type 3 secretion, the main feature is, or the main thing is, bacteria will have like needle like objects in their body, so they will directly invade the cell. So here's the cell, so there is the injected, like they are injecting something here. This is the body part directly from the bacteria, so they're injecting the toxin here. But in case of shigellosis, they don't have it, so they need some sort of way to go into the cell and release the toxin. So they will go to the mucosa through the specialized M cells here, because we know that in the stomach or gastrointestinal tract we have M cells, and because of these M cells here, now the bacteria, so the M cells are helping to go to, like helping the bacteria to get into the cell. So that M cells already helped the bacteria to go into the cell. Here is the bacteria. Now as the bacteria is inside the cell, so bacteria will release the antigen or show the antigen into the cell, and as a result antigen sampling cells will be released from the lymphoid follicles, and antigen presentation leads to the leads of cytokines. Here you can see the cytokines here, and which will recruit more neutrophils here. These are the neutrophils, and these neutrophils will go between the cell junction here. So these are the neutrophils going between the cells, and it will invade the submucosa. So we know this is the mucosal part of the cell, and there is the submucosal part where we have connective tissues or peripheral nerves or nerve tissue bundles, and then these shigella are invading the basal cells here, the basal cells, because already those neutrophils in like attacked that cell junction, so there is more scope for the bacteria to go inside other cells and destroy the host cell. And this is the like way how shigella destroys the cell. Here you can see shigella are multiplying, here multiplying, here multiplying, and going to other cells like the neighboring cells. So from this mechanism we know that shigella needs a secondary way to invade the cell, not like the salmonella or E. coli. They can directly inject their toxins into the cell without any other's help. Here there's another material from the USMLE book, step 1. Here just pause the video and just read, because these are the same thing that I just discussed like up there in the note. So just pay attention to the main features. Now we are at the end of the first part, which is Yersinia. Yersinia has three types: Yersinia pestis, Yersinia enterocolitica, and also in Yersinia pseudotuberculosis. So these are the two reasons of yersiniosis. Here again, the morphology and the factors of pathology and how the pathogenesis occurs. The same thing, they actually multiply in the gut mucosa, and also they causes inflammation or ulceration. So we need to remember that Enterobacteriaceae, they are actually the gut bacteria, so they are most likely to cause diarrhea. There is one interesting thing in about Yersinia is, as we know, the bacteria needs normal temperature, normal body temperature in the human body to multiply. But only in case of Yersinia, here they need 4 to 6 degree of temperature to grow. So the optimal growth occurs during 4 to 6 degree temperature, not in the normal body temperature. So when we will be able to grow or cultivate the bacteria into the lab, we need to keep the petri dish with the agar in 4 to 6 degree temperature box or in the fridge. Here, Yersinia actually transmits from the rodents. It's like an animal, and from the urine, we can, like, if someone is roaming or like doing adventurous thing in the jungle, they are most likely to be affected to the Yersinia, because there can be rodents in the jungle, so from the urine, and also they can be found in the soil. So there is one exceptional thing about Yersinia, that because soils are cold, they're like, whatever weather is this, the soil, they are like 4 to 6 degree or even 10 degrees, so that's the most optimal temperature it can grow. So pay attention to that point. And here I made a last table about clinical symptoms about 4 bacteria, so if you read this table, it will make more easier for you to understand the clinical differentiation between the bacteria. So there is one thing, if we get a clinical case in we don't know how we will identify. So there will be low grade fever for salmonella bacteria, and for the shigella there will be high grade fever, and there is another possibility that the shigella infected patient will have bloody stool. So that's it. And another note I wanted to share, that E.coli is the main reason of urinary tract infection, so you need to pay more attention to that as well. So this is the end of my first part, so let's see the next part.